Okay. Already? Okay. Hi. Well, welcome everyone again to the Edward Ames Seminar here at the Hudson River Foundation. Sorry about that. For those on the, we've got a number of people on the webinar, and for those on the webinar, we have probably about 20 people here in our office, so we thank you all for attending today. You'll notice today is not a Tuesday, it's a Wednesday, and one of the reasons we changed from Tuesday seminars to, we think, a better seminar series, the Net Ames Seminar. Now, I just want to say another word. Our last seminar was the first one after we had renamed the seminar in honor of Ned Ames, who is the former chairman of the board here at the Hudson River Foundation, who has a real keen interest in getting our work and the work of others out to the public. So that's, if you see his name, we're very pleased that we've had Ned to work with the foundation over the years. Today is a lecture of some work that the Hudson River Foundation did not sponsor. However, we'd be very proud if we had. Today we have Dr. Steve Bursian from Michigan State University, and he's here, and I'd like to thank Ike Wergen from NYU Medical Center, who through Ike and his relationship with Steve, we're able to have Steve do kind of a sort of a double program here in New York, one here today at Hudson River Foundation, and Steve will be teaching at Ike's class tomorrow at NYU. The topic today is the effect of PCBs on mink. It's a longer title, which Steve can explain, but Steve is a toxicologist, been a toxicologist for quite some time, has had a real interest in the effects of organic compounds like PCBs and dioxins on, I guess, both domestic animals and wildlife, and a few years ago chose to do some work on mink in the Hudson River. So we thank him for taking some contaminated fish from this area and moving them out to the Midwest. So that was a help, I think. And he's going to report on that work. I think many of you may have read some of his papers, and we're delighted to have him in New York to hear what he has to say. Steve?
Thanks, Dennis. It's a real pleasure to be here and to be able to share some information about the study that we conducted a couple of years ago. Share this information with people that have an interest in the Hudson River. I told Helena, it's nice to finally see this river that provided us our fish that we fed to our mink. As Dennis said, I'm going to be talking about a study that we conducted a couple of years ago where we fed fish collected from the Hudson River, obviously contaminated with PCBs. We fed these diets to ranch mink and looked at the effects on adult reproduction, on offspring, survivability and growth, organ masses, and histopathology. I'd like to just quickly acknowledge the co-authors of this particular study. John Kern and Richard Remington were the ACE statisticians in this study, and thankfully they were involved because this was way beyond my capabilities. Jay Mink is our lab manager and driving force behind everything that we do. And Scott Fitzgerald is a pathologist with the MSU Vet College, and he's been a collaborator on most of our mink studies throughout the years. I'm going to start the talk by giving some general information about the polychlorinated hydrocarbons, and hopefully this isn't information that is going to be too boring for you. I'm sure all of you are very, very familiar with these types of compounds. Then I'm going to focus on those chemicals that resemble TCDD. So we'll spend some time talking about the TCDD like PCBs, dioxins, and furans, which then brings us to the toxic equivalency approach. Since these fish collected from the Hudson River contained dioxins, furans, and PCBs, when we formulated the diets, part of what we took into consideration were the toxic equivalents, the dioxin-like toxicity associated with these fish. Then I'll provide a little bit of information on why we use Mink. We've been using Mink for these types of studies since the late 1960s, and I'll touch a little bit on those early studies, and then I'll specifically talk about the Hudson River study. So the polychlorinated hydrocarbons include the polychlorinated biphenyls, the PCBs, the polychlorinated dibenzodioxins, or the PCBDs or dioxins, and polychlorinated dibenzofurans, or PCBFs, or furans. I'm sure we're all aware that the PCBs, the dioxins, and furans are very significant environmental contaminants. They're found almost everywhere, and in many locations they occur at high enough concentrations that they do cause damage. So they're very widespread, very persistent chemicals that have been out there for a long time. The PCBs and dioxins and furans are often grouped together for a number of reasons. One is they're very similar in structure, and the right-hand column are the structures of a basic PCB congener, a dioxin congener, and furan congener. So in each case, there are two benzene rings attached to each other in various ways, and then there are chlorines attached to the ring. Some of these congeners, PCB congeners, dioxin congeners, and furan congeners, act through the same mechanism of action, and as a result, they cause the same types of effects in a variety of animal species. And the third reason we often talk about them together is because they occur together in the environment. So if you were to collect a fish from the Hudson River or collect a fish from Lake Michigan, you're going to find dioxins, furans, and PCBs in those types of samples. A little bit of background about the PCBs. They were commercially produced in the United States by Monsanto from 1930 to 1975. Roughly 1.4 billion pounds were produced, and the vast majority of those PCBs produced by Monsanto were used here in the United States. PCBs were very stable, heat-stable compounds, and as a result, they were used for a variety of purposes, including non-flammable oils and transformers of condensers, such as pictured here, and this, of course, is very relevant to the Hudson River. They were also used in paints, plasticizers, flame retardants, and they were used in a number of open-ended uses. So consumer products that at the end of their lifetime they were disposed of. So this included insulators and small appliances. They were used as a vehicle for pigment carbonate copy paper, and they were also used in microscope immersion oil. So a lot of uses that resulted in disposal of these compounds over a long period of time. Monsanto ceased production of PCBs for the open-ended uses in 1971, and this was as a result of in the late 60s, it was becoming apparent that these compounds were being found in places that one would not expect them to be found. And studies with rodents and primates were indicating that these compounds were having very deleterious effects on animals. So this prompted the banning of these compounds, and finally in 1977 they were banned completely. It's estimated that 54% of these compounds are still in use, and that 31% still exist in the environment as contaminants. Monsanto's commercial mixtures were sold under the trade name of Aerochlors, and there were 50 Aerochlor products. Typically, the term Aerochlor was followed by a 4-digit number. Common Aerochlor mixtures that are given here, Aerochlor 1254, 1260. There were a number of these commercial mixtures that were produced. And as I said, these are mixtures, so these commercial products contained a vast number of individual congeners. So this is a generic PCB molecule, 2 benzene rings stuck together through the 2 carbons, and there are 5 binding sites on each of the rings where one can attach a variable number of chlorines. So given the variable number of chlorines and the different positions where these chlorines can be attached, there are 209 possible PCB congeners. Just talking briefly about the dioxins and the furans, these compounds obviously were never intentionally produced, rather they were byproducts of a variety of processes that included leaching of paper by pulp and paper mills, incineration of waste, burning of wood in fireplaces. They were also released during refining of petroleum and also during the synthesis of a variety of chlorinated compounds such as chlorophene, chlorophenol, and of course the phenoxy herbicides that are associated with Agent Orange. So this is a generic dioxin congener. Again, 2 benzene rings connected through 2 oxygens. There are 4 binding spots on each ring, and each binding sites are numbered. Again, given the different chlorines or the different number of chlorines that can be attached, and in the various positions, there are 75 different congeners that are possible. This is a generic furan congener. Again, 2 benzene rings attached to a furan moiety. Again, 4 binding sites on each ring, number 1 through 9, is pictured here. And there are 135 different furan congeners that are possible. So that brings us to the comparison of these various congeners. Of these 209 PCB congeners, the 75 dioxin congeners, and 135 furan congeners. The one that is considered to be the most toxic is the molecule here. I'm sure everybody's familiar with 2378 tetrachlorodibenzoyl or TCDD. And you can see that I've highlighted the chlorines in red. So there are chlorines attached on carbons 2 and 3, 7 and 8 in TCDD. So these chlorines are on the outside of the ring. We take a look at the PCB congeners. Of the 209 possible PCB congeners, the most toxic are the non-ortho coplanar PCB congeners. I've pictured a generic PCB congener here. And as the name implies, a non-ortho PCB congener does not have chlorines attached in the ortho position in carbons 2 and 6. So rather, in these congeners, the chlorines occur in carbons 3, 4, and or 5. So I highlighted those binding sites in red. So like TCDD, those chlorines are on the outside of the ring. And there are 4 non-ortho coplanar PCB congeners that are important. And I've highlighted PCB 126 in red. 1, because it's the most toxic of the non-ortho PCB congeners. And 2, it's the driving force behind the results that we've seen in the Hudson River study and also other locations such as Saginaw River and Saginaw Bay in Michigan and Housatonic River in Massachusetts. It's PCB 126 that seems to contribute most of the toxicity associated with feeding fish. Monoortho PCB congeners are PCB congeners that have, as the name implies, one chlorine in the ortho position. So there's a chlorine attached to either carbon number 2 or carbon number 6. As a result of that placement of the chlorine, these rings have to rotate a bit to accommodate placement of that chlorine. And as a result, these monoortho PCB congeners are not nearly as toxic as the non-ortho PCB congeners because of this slight rotation. There are 8 mono PCB congeners that are similar to TCDD in terms of toxicity. Looking at the dioxins, there are 7 dioxin congeners that resemble TCDD, and there are 10 furan congeners, isomers that resemble TCDD. So in total, we have 29 congeners, PCB congeners, furan congeners, and dioxin congeners that resemble TCDD and possess some degree of TCDD toxicity. As a result of the similarity in structure, these dioxin-like or TCDD-like congeners have the same mechanism of action, as I said, they cause the same types of effects. So TCDD binds to a cytosolic protein called the aryl hydrocarbon receptor, the AH receptor, that dioxin receptor complex then migrates to the nucleus of the cell. Sequence of events occurs, and this ultimately results in toxic effects that we commonly see in animals. The TCDD-like PCBs, dioxins, and furans also bind to that AH receptor, and as a result cause the same types of effects that TCDD does, although they're not nearly as potent as TCDD. Which brings us to the toxic equivalency approach. So this is an approach that can be used to normalize the toxicity of these various congeners, because as I said, typically in a sample of a fish, for example, we're going to see all 3 classes of compounds. So it's advantageous to try to figure out the total TCDD toxicity associated with that sample. So we use a toxic equivalency approach. And we can do so, again, because they all have, these TCDD-like chemicals all have the same action. So what has occurred is that various in vitro studies and in vivo studies have been conducted over the years, and the toxicity of these individual congeners has been assessed and compared to the toxicity of TCDD. And as a result, each one of these congeners has been assigned a toxic equivalency factor, or TEF, value that reflects each congener's toxicity relative to TCDD, which is given a TEF value of 1 because that's considered to be the most toxic. So in a situation where, for example, we're feeding contaminated fish to mink, not only do we want to know the total concentration of PCBs, but we also want to know the total amount of dioxin or TCDD-like toxicity associated with those fish. So what we do is identify the 29 TCDD-like congeners in that sample, and then multiply those concentrations by each congener's toxic equivalency factor to give us the toxic equivalence or dioxin-like toxicity that is contributed by each one of those congeners. And we add those up. We add the TEQs contributed by each of the congeners to get the total dioxin-like toxicity associated with that fish. I'm going to give an example here in just a second. Let me just quickly go through the TEF values. These are TEF values for the various dioxin congeners. I've pictured TCDD in red because As I said, that is considered to be the most toxic, and it has a TEF value of 1. Notice also that 1, 2, 3, 7, 8 pentachlorodibenzodioxin also has a TEF value of 1, so its toxicity is fairly similar, just TCDB. The other dioxin conjugates are somewhat less toxic as reflected by their TEF values. These are the furan, TCDD like furan congeners and their TEF values. I've highlighted 2, 3, 4, 7, 8 pentachlorophenol, or pentachlorodibenzophuran in red because that is the most toxic of the furan congeners, and oftentimes has a fair amount of contribution in terms of dioxin-like toxicity in fish depending upon where they're collected. PCB congeners, the non-ortho PCB congeners, again PCB 126 highlighted in red because that is the most toxic, TF value of 0.1, meaning it is approximately 1/10 as toxic as PCB, so it takes 10 times more of the compound to cause the same severity of effects that one typically sees with PCB. And then finally, the monoortho PCB congeners, they all have been assigned a TEF value of 3 times 10 to the minus 4, which reflects their very limited toxicity. However, they tend to occur in the greatest concentrations in the environment, as we'll see here in just a second. So I've included this table just to illustrate how we use this information. So we'll take a fish from Lake Wobegon, and the column on the left just indicates the various dioxin-like or TCDD-like congeners detected in the fish. PCB 126 and 169 are two of the non-ortho PCB congeners. PCB 156 is a monoortho PCB congener. Then we have TCDD and the RAM. Next column over gives the concentration found in the fish, and notice that PCB 156, the concentration is very, very high. And again, this is something that we typically see in fish, very high concentrations of these monoortho PCB congeners, at least some of them. Next column over gives the TEF value of each of the congeners. And then finally, the last column illustrates the toxic equivalents contributed by each one of those congeners. So for PCB 126, we simply multiply the concentration by the TEF value, and we see that PCB 126 contributes 41 picograms of dioxin toxic equivalents in this sample of fish. We add them all up, we see that all of these congeners combined provide about 48 picograms of TCDD per gram fish, and that PCB 126 contributes the vast majority of that toxicity. Most of those TEQs are provided by PCB 126, and that's what we've seen in the Hudson River study, as well as the Houstonic River, where fish are contaminated with PCBs in a situation very similar to the Hudson River. So a few words about why we use Mink in these types of studies. For the past several years, Mink have been used in a variety of studies to represent fish-eating mammals, piscivorous mammals. These studies actually began way back in the late 1960s. A retired colleague of mine and a good friend, Dick Ulrich, and his mentor, Bob Ringer, were one of the first people to conduct these Mink studies back in the late 60s. Commercial fur ranchers surrounding the Great Lakes were utilizing Great Lakes fish as a cheap protein source. And during this period in the late 60s, the Mink ranchers started reporting significant reproductive problems on their ranches. So the females either were not delivering kits, they weren't whelping, or if kits were born, young mink, if kits were born, they weren't surviving very long. So obviously something was going on. They very quickly figured out that fish was the problem. The initial thought was that it was DDT in the fish. So Ulrich and Ringer fed various concentrations of DDT to Mink and found absolutely nothing. And this was also at the time where studies related to PCBs, the air core mixtures, were being published. So then they decided to try feeding a couple of different air core mixtures to Mink, along with fish collected from the Great Lakes, various species of fish from the Great Lakes. And they were able to duplicate the reproductive problems that were being reported by the ranchers. So this is really the first time that one had identified the PCBs were a problem in Lake Michigan. Fish were heavily contaminated with these compounds, and that mink were very sensitive to these types of compounds. So since the late 60s, mink have been used in a variety of studies dealing with the effects of PCBs. And now agencies such as the Fish and Wildlife Service and EPA have identified the mink as the canary in the coal mine. If something is going on in mink in a particular location or doing a site-specific study, then it certainly is a red flag that there might be trouble. So the mink typically is exposed, depending upon the location, to relatively high concentrations of these compounds, primarily because of its diet. Mink do eat fish. The percentage of fish in the diet varies depending upon what's available. And also, because of their position in the food chain, animals are carnivores, so because they're at the top of the food chain, because of the bioconcentration, the compounds or the concentrations of these compounds found in mink are relatively high. As I pointed out, Ulrich and Ringer first demonstrated that mink are very, very sensitive to these compounds, to these TCDD-like compounds, second only to the guinea pig. And they can be used in a controlled exposure situation if one happens to have a mink facility, which we're fortunate to have. So it's much easier to get a handle on how these compounds are affecting mink, obviously in a farm situation compared to trying to figure out what's going on in the wild. So that leads me to the Hudson River study that was funded by Fish and Wildlife Service and the trustees of the Hudson River. This is part of the Natural Resource Damage Assessment. Provides a little bit of information, and I'm sure you're all familiar, probably more familiar with this than I am, but the Hudson River is contaminated with PCBs for hundreds of miles from Fort Edward to the city. And the historical source of these PCBs were manufacturing facilities located at Fort Edward and Hudson Falls. Field studies conducted along the Hudson River over the past 30 years have indicated that concentrations of PCBs in the livers of these crab mink are high enough where they're relatively close to concentrations found in ranch mink that have been associated with reproductive problems. Bob Foley and Associates did one of the initial studies 30 years ago, and then Dave Mayak and Associates did a more recent study indicating that the concentrations of PCBs reported by Foley have not gone down that much in the subsequent 30 years. So taking a look at the latter study, mink were collected from PCB-contaminated sections of the Hudson River between Fort Edward and Troy between 1998 and 2001. And they determined the concentrations of some PCBs in the livers of these animals, and they found that animals collected within 6 kilometers of the river, which is equivalent to roughly 1 home range, those concentrations were 13 micrograms per kilogram lipid. And those trapped within a kilometer of the river had higher concentrations. So their concentrations of PCBs and milliliters were on average 33 micrograms total PCBs per gram liver. So comparing these concentrations to concentrations in livers of ranch mink that were fed diets containing contaminated fish, these values are fairly close. So Sylvia Heaton, graduate student of ours back in 1995, conducted a fish feeding study. Fish were collected from Saginaw Bay, Michigan, and there were a number of automobile facilities located on the Saginaw River that contributed PCBs. So we conducted that study very similar to the Hudson River study. Fed mink diets containing these PCB-contaminated fish. And she determined in her study the lowest observed adverse effect concentration. So that concentration in the liver, the lowest concentration PCBs in the liver associated with the decrease in kid survivability was 45 micrograms per gram. And then in the study that we conducted in 2006, related to the Hudson River in Massachusetts, heavily contaminated with PCBs as a result of the similar industrial activities as the Hudson River, we found that the lowest concentration in the liver associated with reduced kid survivability was 29 micrograms per gram. So the levels of PCBs found in the livers of these trapped mink on the Hudson River are very close to the levels in the livers our ranch mink exposed to these compounds associated with reproductive problems. So based on that information, at least in part, we were asked to conduct this study. And the objective of the study was to evaluate the health effects of eating ranch mink diets containing various quantities of PCB-contaminated fish collected from the Hudson River. We looked at reproductive performance of the adults. We looked at offspring survivability and growth. We looked at organ mass and tissue pathology in these animals. So carp were collected in 3 different locations on the upper Hudson River between Fort Edward and just south of Mechanicville. Those 3 locations are pictured here. Specifically, they were Moses Kill, Northumberland Pool, and then in the vicinity of Block 2. So we had about 3,000 pounds of carp that were collected by the New York Department of Environmental Quality. And they used an apparatus such as this. A shocking boat, so electrical currents that's generated in the water that create this sponge that fish they float to the top and netted out of the boat, netted into the boat. So, like I said, about 3,000 pounds of carp were collected for us. They were shipped to us frozen. They arrived midnight one night. Poor guy from the NYDEC had to drive a long distance that night to deliver us our fish. We put them in a locked walk-in freezer until we were able to grind fish into incorporate them into our mink diets. So this shows a box of the fish each. One of those carp was individually packaged in a plastic bag. It had a tag on it that indicated where it was caught, the length, the weight, and the date of capture. So as I said, these fish were collected from 3 different locations. And as we were grinding and mixing them, we had to record what fish were being ground in what mix. So these 3,000 carp, the 3,000 pounds of carp, we had to grind them in three different grinds. So for each grind, we took a proportionate number of fish from each location ground that, labeled it grind 1, and did the same for grind 2 and grind 3. And then when we incorporated that fish into the diets, then we took an equal amount of fish for each grind. So we ensured that those diets contain a good proportion of fish from each of the 3 locations. So ground fish, we have a big grinder, and it comes out looking like hamburger. We ground the fish. Obviously, the Hudson River fish was a treated fish, and then we used ocean herring as our control fish. So the total incorporation, the rate of total fish incorporated into the diet was 20%. So the control diet contained 20% ocean herring. High dose of PCB contaminated diet contained 20% of the Hudson River fish. Picture was taken a number of years ago. A typical diet used on commercial farms is a wet diet. So in this particular case, it consisted of 20% fish. And then to that, we add ingredients such as a mink cereal. It's a cereal specially formulated for mink, along with dried egg, dried liver, dried blood protein, vitamins and minerals, water, and oil. So we end up with this kind of wet hamburger type stuff that we feed daily. The other thing we had to do was, because these fish contain thiaminase, which breaks down thiamine in the diet, and mink are very susceptible to a thiamine deficiency. In order to prevent this thiamine deficiency, we had to go through every morning and give them a little dollop of feed containing thiamine, an excessive amount of thiamine, to try to counteract the effects of the thiaminase in the fish. Years ago, when we were doing these fish studies, we cooked the fish first because heat inactivates the enzyme. We ran into, the last time we did it, we ran into a problem because we were cooking this fish in a room underneath MSU Dairy Store, and we ended up ruining a batch of their ice cream because it tasted fish. So that ended that. And we were also criticized that perhaps cooking fish is not realistic for an animal in the wild. So then we went to using fresh fish and feeding them thiamine. So this table illustrates the different dietary concentrations. So as I said, the total amount of fish incorporated into the diet is 20%. A mink in the wild will eat anywhere from 0 to 50 to 60% fish, depending upon the location. So you can see our control diet contained 20% ocean herring. The high dose diet contained 20% Hudson River fish, and the doses in between contain various combinations of the two. So our targeted dietary concentrations, based on the average PCB concentration in that fish that we received, the average concentration being 36 parts per million PCBs, or 36 micrograms PCBs per gram fish. We formulated these diets in such a way that our targeted concentrations range from 0.9 micrograms per gram diet up to 7.2 micrograms per gram diet. The analyzed dietary concentrations, which were the concentrations that I'll use from this point forward, and those are the concentrations used in the manuscripts, the papers published, they were fairly close to what our targeted concentrations were. So the analyzed concentrations range from 0.72 to 6.1 micrograms total PCBs per gram diet. And then the total toxic equivalents, the TCDD-like toxicity associated with these diets, range from 5.4 to 38 micrograms of TCDD toxic equivalents per gram diet. So, contribution of the different components contained in these fish to the PEQs, the dioxins and the furans not a whole lot there. So the dioxins and the furans each contributed about 1.5% of total TBQs. The PCBs contributed 97% of the dioxin toxicity. And of that, the non-ortho PCBs contributed by far the vast majority, 75%, and all of that was essentially PCB 126. So again, PCB 126 was the driver of this. Mono-ortho PCBs contributed 20%. Very similar to what we've seen in other studies, particularly the Housatonic River. In the Saginaw River in Michigan, we tended to see more furans and dioxins than here, but typically PCB 126 is the content that drives all this. So this table just briefly it indicates the number of animals on our various treatment diets based on advice from two statisticians. So the control group and the two high-dose groups contain 15 females, and the in-between doses had 10 females in the group, and then all dose groups had 5 males. And we fed the males along with the females. We were fed the same diets. So our animals were housed in the house singly in an open-sided pole barn. This is a picture of the shed that the study was conducted in. This resembles a typical commercial mink ranch, although on a much smaller scale than you would typically see in a commercial operation. But the interior looks very similar. We fed the test diets beginning 8 weeks prior to breeding. Mink breed once a year from March 1st to March 21st. So you get one shot a year to do a reproduction trial. So in this particular case, we started feeding the diet on January 3rd. We continued feeding those diets through the weaning of the kits. We typically try to wean kits around 6 or 7 weeks of age. This is in the neighborhood of mid to late June. So those adult females were on the treated diet for approximately 160 days. When kids are born, we typically, what we do is go through every morning and listen for the squeaking of kits, and if we hear a new noise in a nest box, flip their ID card over, and then we'll go back 24 hours later and weigh them, determine the gender and weigh the kits. We found a long time ago if we interfere with the process before 24 hours, oftentimes the mom will lose the kits. Didn't want that happen. And then those kits were weighed again at 3 and 6 weeks of age. At the time that the kits were weaned, again when they were about 6 weeks of age, we terminated the adult animals, both females and the males, and we took a portion of the 6-week-old kits and biopsied those. We took out a variety of tissues. Listed here, liver, brain, heart, kidney, spleen, thyroid, adrenal glands, testes and uterus, the mandible and maxilla, which I'll talk about later here, several slides down. These tissues were weighed and then fixed for evaluation by Scott Fitzgerald at Pathology. We also froze a portion of the liver for subsequent contaminant analysis, both total PCBs as well as the contaminant analysis. So the kits that we had left over after we sampled the 6-week-old kits for the necropsy at that point in time, the remaining kits were kept on their respective diets until they were approximately 31 weeks old. So this trial ran from the beginning of January until the end of November. And this is typically when a mink is pelleted, late November, early December. This is considered the end of their growth period. And I've listed the number of kits carried forward that were kept on their diets through 31 weeks of age. And you know, the last 4 dose groups are in red, and that is because those kits did not live for 31 weeks. At various points along the way, they died. So at the end of that 31-week period, we just had kits left in the control group and kits left in the low-dose group. So at the end of that 31 weeks, then we necropsied 30 controls and the remaining 23 juveniles in the low-dose group. Took the same tissues out, did the process in the same way. This table simply illustrates the various endpoints that we examined during the course of the study and the various types of statistical analyses that David and Richard did, or John and Richard did. So basically, we looked at adult body weight and adult feed consumption. We looked at a variety of reproductive parameters, including the number of females that mated, the number of the gestation length, the number of females that had litters, litter size, average litter weight, and then kit body weight at birth, 3 and 6 weeks of age, kit survivability at birth, 6 and 3 weeks to 6 weeks of age, and then monthly body weights of the juveniles through their 31-week period. Organ weights in all 3 age groups, PCB concentrations and toxic equivalents, concentrations in the liver in all 3 age groups, and histopathology of the various tissues, concentrating specifically on the claws. So to quickly go over the data, this is a bar graph simply illustrating the number of females that actually delivered kits. And despite the fact that there were no significant differences, you can see that at the 3 top doses, we're beginning to see a decrease in the number of females delivering offspring. Again, as I said, these differences were not significant. This graph illustrates the number of live kits that were born with each litter. And you can see at the 2 high dose groups, there was a significant decrease in the number of live kits. So obviously, exposure to these compounds in utero is having an adverse effect on the development of fetuses. This graph illustrates the percent survival of 6-week-old kits in the top 3 doses, caused a dramatic decrease in survival of these offspring. And this is typically what we see. In these mink feeding studies, if a female delivers kits that have been fed PCBs, typically this 6-week period, and of that 6-week period, particularly the first 3 weeks, are really critical. So we see mortality is generally going to occur in the first 3 weeks and in the first 6 weeks, which was a bit of a surprise in this trial because we continued to have animals die throughout this trial. So, kit survivability affected by the top 3 doses through 6 weeks of age. Growth of the kits through 6 weeks of age was affected by exposure to PCBs, so all of the lowest dose of PCBs had an effect on these animals. Notice that in the males, we don't have a mask for the high dose group, and that is because there were only 2 kits from that high dose group that were still alive at 6 weeks of age, and both of them. And there's certainly an effect on body mass. Now, typically, if at 6 weeks of age, if these animals are slow or small, they will continue to be small for the rest of their lives. They simply will not catch up. This bar graph illustrates offspring mortality between 6 and 31 weeks of age. It's kind of a busy graph, but you can see that only in the control group and the low dose group of 0.72 micrograms PCBs per gram feed did we have kits surviving the full 31-week period. In all of the other groups, those kits died before we reached 31 weeks. in the last 2 dose groups, those kids died before we hit 10 weeks. So, and as I said, this is somewhat unusual. Typically, if animals can make it through the first 6 weeks, they're going to survive. They may be small, but they'll survive. In this case, we had animals continuing to die throughout the trial. This table shows the concentrations of PCBs in the diet as well as in the liver. So if you take a look at the numbers in black, top row would be the dietary concentrations of PCBs. If you look at the second row of numbers in black, these are the concentrations of PCBs in the liver of the adult females. And you can see that the dietary concentrations correspond fairly closely to the concentrations of PCBs we see in the liver. But if you look at the concentrations of TEQs in the feed, top row of blue numbers, comparing it to the bottom row of blue numbers, you can see that the concentration of TEQs in the liver are considerably higher than what is present in the feed. So obviously what is happening is that these animals are retaining these PCB-like congeners that contribute to the dioxin-like toxicity. They're retaining them in the liver, which could account for this mortality that we see. So there is some bioaccumulation in these compounds. The next 4 graphs are Dave and John and Richard modeled the survivability data to come up with lethal concentrations, LC20 values. So by modeling the data, they use methodology similar to the EPA benchmark modeling procedure. We modeled the data in such a way that we were able to derive a dose at which we would expect 20% of the population to experience mortality. So this very faint line is the actual LC20 values. So based on dietary concentrations of PCBs, the LC20 value is about 0.3 micrograms PCBs per gram feed. This graph illustrates the LC20 value based on dietary TEQ values, and roughly a dietary concentration of 3 picograms TEQs would be expected to result in 20% mortality of 6-week-old kids. LC20 values based on concentrations within the liver. So the LC20 value here is about 0.8 micrograms PCBs in the liver, corresponds with 20% mortality in the kids. And a concentration of about 13 micrograms TEQs in the liver corresponds to 20% mortality. Now, we decided to express the data this way. Fortunately, we had enough data points where these data could be modeled. We felt that it was perhaps a more accurate depiction of what was going on rather than simply present OAL and OAL values, particularly when one is trying to compare studies. And that's oftentimes all we have. We simply compare an OAL value across 2 or 3 studies. And those no AL values and the low AL values reported by individual studies, of course, depends upon the doses that the experimenters choose to use for that particular trial. So by using this methodology, it might be a little bit easier to compare across studies and get a better idea of concentrations causing the same degree of effect. We were able to do this in the Hudson River study, so in that particular case, we were able to compare LC values between these studies. The next 3 slides just show differences in organ mass. So we had an increase in thyroid mass to 2 high doses in the adult females. There should be an asterisk on the last bar. So both both the 2.8 and 6.1 dose resulted in an increase in thyroid mass in adult females. We had a decrease in heart mass in both 6-week-old males and 6-week-old females. And there was, again, with the same asterisk here, but we had an increase, a significant increase in adrenal gland mass in both female and male juvenile males, so Mink that were 31 weeks old. We don't have a good idea of what this means. The pathologists in examining these tissues indicated there were no pathological changes in these tissues, so I'm really not sure what the biological significance of this increase in organ weight is. But we reported it. Okay, now I'm going to talk about a jaw lesion that we first saw in Mink, I don't know, 10, 15 years ago. We kind of discovered it by accident. We were trying to determine whether or not PCD 126 would cause a change in the weight and structure of the penis bone of the Mink baculum, and we ended up didn't do anything to the baculum, but it causes some jaw lesion, and I'll show you pictures of that. So ever since we saw this jaw lesion in Mink, we started looking at it at every Mink study we've done, and we found it in every Mink study we've done, including the Hudson River study. And again, I'll show you some pictures of what is going on. But this table illustrates the concentration of PCBs in both the feed and the liver associated with a 20% incidence of this jaw lesion. So looking at dietary concentration, the concentration of 2.3 micrograms total PCBs, and a concentration of 15 picograms TQs is associated with incidence of this lesion in 20% of the Mink. Hepatic concentrations of 2.8 micrograms PCBs and 89 micrograms TQs. So the next 4 slides are slides of cross-sections of jaws, just to illustrate what is going on. This is a maxilla from a control animal, this cross-section through the jaw. And the T refers to teeth, so you see these round structures are the teeth. This arrow, the PL, refers to periodontal ligament, and actually this arrow should be around the tooth, so it's the ligament that is surrounding the tooth. And then the rest of this pink area in here is the alveolar bone. So this is the jawbone that's anchoring the teeth. This is a normal situation. This slide is a cross-section of the maxilla from an animal on the next to the highest dose, 4.5 microgram dose. So here you can see the tooth. And what should not be there is this area labeled SCC, it's squamous cell 6. So what we think is happening is that exposure to these compounds is causing a proliferation of squamous epithelial cells. We think from the gums, I'm not sure. There's a proliferation of these cells inward, forming these cysts, and as these cysts appear, they're eroding the bone. So the higher the concentration of PCBs these animals are exposed to, and the longer as time of exposure increases, the number of these cysts increases. So another cross-section from an animal on that next to the highest dose. Again, as you can see that in this case, we've got a rather large area of these squamous cell cysts. And again, that area occupied by those cysts is an absence of bone. And then finally, this is a cross-section of a jaw from an animal on a high-dose group. Again, here's the tooth. And then these, the squamous cell cysts are located all the way around the tooth. And again, that corresponds to an absence of bone. Now, we did not see this in this particular study, but if an animal is exposed to a high enough dose, or if enough time passes, you see something like this grossly. So I don't have a picture of a control animal. But what happens first is that there's a misalignment of the teeth. So in this animal here, these teeth should be very close together, very tightly arranged. And you can see that there's beginning to see a bit of a misalignment in these incisors. And then as time progresses, these teeth become more and more misaligned. And then finally we get a situation where these teeth fall out. There's no bone left to anchor the teeth, and this is not a good thing for a carnivore. So we're assuming that in the wild, experiencing this type of condition, chances of survival are pretty remote. So to conclude, the study indicated that the reproductive performance of adult female mink and offspring survival and growth were adversely affected by consumption of feed containing PCBs derived from fish collected from the Hudson River. And we estimated, based on the average concentration of fish collected from the Hudson River, not the contaminated fish we use, but rather just fish collected in the river, that we were seeing effects on survival equivalent to concentrations equivalent to a mink consuming 10% fish each diet. So that's kind of on the low end of the amount of fish a mink typically consumes alive. So specifically, we saw stillborn kits, or an increase in stillborn kits per litter at concentrations of 4.5 micrograms PCBs and above, corresponding to 28 micrograms of PDQs. The LC20 value for kit survivability based on the diet was 0.34 micrograms PCBs per gram feed. The hepatic LC20 value was 0.8 micrograms PCBs per gram liver. And then in terms of kit growth, kit body mass was significantly decreased at doses of 1.5, 2.8, 4.5 micrograms PCBs. Organ masses, we just went through those. Thyroid masses of females were increased, and as well as the gland masses of the juvenile mink, heart masses of 6-week-old kids were significantly increased. In terms of tissue histopathology, the dietary EC20 value for the jaw lesion was 2.3 micrograms PCBs, and the corresponding concentration in the liver was 2.8 micrograms PCBs. So this study was a little bit different compared to the Housatonic River study. In the case of the Hudson River study, the effective concentration for the jaw lesion was 6-fold greater than the concentration that resulted in a reduced kit survivability. In the Housatonic River study, it was exactly the opposite. We saw the jaw lesion at lower concentrations than concentrations that resulted in kit survivability. I'm not sure why, but that's so, the disclaimer, these conclusions are mine, or the authors, and they do not necessarily reflect the position of the business. I'm happy to take any questions.
Have reduced heart disproportional with the weight increase as well?
Yes, that was relative. Well, I should have said that. That was relative heart mass. So corrected for body weight.
You had all these dying kits.
Yeah.
Did you do any histopathology or post-mortem, anything to try to figure out exactly why they died?
That's a very good question. Unfortunately, in most cases, when a kid dies, mom eats it. So either they disappeared, they weren't there, or we saw bits and pieces. Now, what we think might be happening is studies with rodents have indicated that exposure to TCDD can influence development of the mammary gland. We haven't tested this in the mink, but we're wondering if perhaps the amount of milk produced by mom or the quality of the milk is not sufficient to maintain growth.
Okay. A follow-up. I understand that the non-PCBD like congeners of PCBs, at least some of them, tend to be neurotoxic and affect behavior. And is there a possibility that a mother's behavior could have been altered and resulted a greater percentage of the kid's time?
That is another excellent suggestion, and we have not specifically looked at that. And certainly, just casually observing these females, you can very quickly tell a good mom and a bad mom. And typically, moms that don't do a good job of raising kids, they are disinterested. The kids may die of hypothermia at night because they're not being cuddled and nursed. And so that's a good point. It could be a behavioral effect of this kind.
Do you have to note amongst the three locations where you were collecting fish, whether it's variation like concentration or well?
Yeah, and I can't remember. I'd have to look at. I brought the paper with me. I'd have to look at the paper to see. But yeah, one of the, I think it was your couple the fish from that area, I think had high concentrations. I think there was a twofold difference in concentrations of the three sites.
Do you have to have any considerations on possibly affecting climate cholesterol, affecting behavior, effect of eliminating toxins, the stressors being on living, possibly increasing toxin storage as a result of the stress living and changing behaviors of mothers approaching the wild?
For me, I don't know. That's a big point. And I think Ike and I were talking about this last night. It's interesting. We've seen this in a couple of studies where we do these site-specific feeding studies and show effects, and then field biologists go out and they don't necessarily see the same. Yeah, there may be something about that compliance situation that makes us.
Yeah, it's affect toxin removal and conversion adding.
They're quite good points.
Back to behavior, really is opposite suppression variables.
Yes, very, very, very much. Typically, we look at feed consumption during from the beginning of the trial until we start breeding the females, and then we stop, so we're not disrupting them. In this particular case, interestingly enough, the animals on those diets containing greater concentrations of Hudson river fish actually ate better than the control animals. It wasn't significant, but it's not unusual for the other way around, that higher concentration from those schools. And that could contribute on how quality or not quality.
You mentioned in the beginning that well, if fish is available for it, but are there any particular being shown for fish in highly teaching tolerance for children? Or do they just eat it?
Is that occur in the wild?
Yeah.
I'm not sure. I don't know. And like I said, in this particular case, the mink like the Hudson River fish better than the So you know, typically ranch mink, once they get used to a certain feed, if you change some ingredient in that, it can be pretty tender. There's always a concern. I It could be that there are enough differences in the case.
I don't know anything about the mink, but I was kind of curious about what the gestation period was, whether they mating, did you mate these before injected, before they started feeding PCBs, or well after?
We start them in January, and then they're mated in March. So they're on feed for about 2 months before we mate them. Mink have a delayed implantation, so we determine gestation period as the time between, let me back up a little bit. On the ranch, we try, during this 3-week period, we try to mate a female with 3 or 4 different males. The more males, the better. So the gestation length we determined to be the time between mating, the last successful mating of the last male to the time of delivery. So average gestation length is around 48 weeks. And sometimes we've seen changes in gestation length as a result of exposure to these compounds.
And they were feeding 2 months prior to that?
They started on their treated feed 2 months prior to me.
The females on a particular dose level were mated with males on the same
dose level. Yeah.
I know you gave the vitamin supplements. Do you hear a difference in the vitaminase, the hearing, and the climate? Did that be a down factor, or did the supplements making all that out?
We felt that we gave enough thiamine that it would even all of that out. As I said, mink are very, very sensitive to a thiamine deficiency, and they develop what's called JAS6 paralysis. And they very quickly, within a matter of days, will develop this thiamine paralysis that indicates a thiamine deficiency. And we did not see that, so we felt comfortable that they were getting enough on it.
I'm hoping to do a follow-up on the lower doses where you have kids survival to grow them up to see how it's their reproduction. Grow them up on a clean diet, to see if there is an effect of the early life exposure when they were in utero.
Right.
And see about their reproductive situation.
Well, years ago, one of our grad students, Janelle Reston, did a similar study. We fed concentrations of PCBs up to one part per million. And then she took that first generation offspring. Some continued on treated feed, some were put on a clean feed. They were all kinds of mating combinations. And she did find that in the second generation, there were animals where reproductive performance was adversely affected. Relatively small number of animals, and there were relatively few doses, so this is something that probably should be repeated, but that was at least a hint. There might be an effect in generation to generation.
So yeah, I noticed that you have significantly smaller hearts. Treated animals, controls. With fishes, the hearts are the primary target in terms of almost all the toxicity. Did you notice any difference in terms of cardiovascular function, or did you look for it in treated animals versus controls?
We did not look for it, and after we talked about that, I'm aware that same thing happens in birds exposed to these compounds. There are changes in heart morphology and performance. So that would be something to look at down the road, maybe a small number of animals exposed to compounds and focus specifically on that. Make her a little difficult to work with, to do things that you do with other species. They're not the friendliest critters in the world.
This fish may have had an accumulation of other toxins, like mercury and
right.
And so this cumulative effect may have had a greater impact on the fish, on the mink.
Exactly. So a number of years ago, when we were firebombed by PETA for doing this kind of work, because we discovered in the 60s PCBs did adverse things to Mink, so why are you still doing it? But at any rate, that brings up the point of these site-specific studies. Even though PCBs may be the predominant contaminant, you're exactly right. There are a lot of other things in that case. So that's why I think we see differences between the Housatonic River study and the Hudson River study and the Saginaw Bay study. There are differences in these other components. We looked at heavy metals, mercury, chlorinated insecticides, polybrominated diphenyl ethers in these fish. The concentrations of all these compounds we felt were below concentrations that would result in effects by themselves. But you make a good point. We really don't know what synergistic effects or additive effects these compounds can have. And that's why I think these site-specific studies are important. There are differences between sites because of this.
Right. So in the past, you looked at the toxicity of PCB 126 alone.
Right.
Correct. So is there any difference that you see in terms of toxicity of 126 versus the contaminated species in terms of endpoints concentration?
I think generally speaking, in doing a fish feeding study like this, you see effects at lower concentrations when you do feeding the pure chemical. And it could be for any number of reasons. I mean, you suggested behavioral effects from these monoorthic PCB compounds. Other chemicals could be causing the effects compared to a pure chemical. What would be nice to do, and we've talked about doing this, but we're always limited by money, would be if we're doing a fish feeding study to also run a PCB 126 groups at the same time at different doses to be better able to compare.
Has anybody gone out in the wild besides the Hudson, Great Lakes, look at what the population effects are?
Yeah, we did a study for Dow Chemical about 5 years ago where we administered pure compound, a tetrachlorofuran and a pentachlorophuran and TCDD to Mink, and we found the typical reproductive effects of jaw lesion effects. And then colleague Matt Zwiernik did a field study along the Tittabawassee River, and Mink was flourishing, in other words. So what we found in the lab was not necessarily corresponding to what he was finding in the environment. I'm a misreader sometimes.
Do you have any thoughts on that?
I didn't have a question.
Well, what you find in the field versus the laboratory?
I think it's different from that.
Yeah. No one on the webinar has asked any questions yet. Any more questions here? Well, I have to say, that was a terrific chemistry lesson. I think a lot of us would have loved to have seen your explanation of congeners and non-planers and so forth. That's 20 years ago, but they don't like it. So I thank you for that. And again, I want to remind everyone again, we've got a lecture coming up first week of December. It is a Tuesday. Not sure whether that's the second or the first. So Rico Mullen from the college is going to be here talking about some pathogens in sediments and in air in the harbor. So hope to see you then. Again, thank you, Steve.
Thanks for the invitation.